Today we will be talking about osmolarity and comparing it to tonicity. To do that, we will look at three different beakers containing solutions of different concentrations. Osmolarity can be used to describe either the solution or the cell. Each dot I draw in the beakers will represent 100 osmoles. So the first solution will be a 300 osmolar solution. The second will be a 100 osmolar, and the third will be a 600 osmolar solution. We will compare each solution to a cell with a 300 osmolar concentration. When talking about osmolarity, we can compare cells and the solutions even when they are not in contact with each other. Because the solution in beaker 1 and the cell have the same concentration, the solution is isoosmotic to the cell and the cell is also isoosmotic to the solution. Because the concentration of the solution in beaker 2 is less than the concentration of the cell, the solution is considered hypoosmotic to the cell and the cell is hyperosmotic to the solution. In beaker 3, the solution is more concentrated than the cell, so the opposite terms would be used to describe the comparison. Osmolarity can be used to describe the cell and the solution apart, but tonicity describes what happens to the cell when placed in the solution. Remember, tonicity always describes the solution, not the cell. When the cell is placed in the equal concentration first solution, nothing happens to the size of the cell. This is called an isotonic solution. When the cell is placed in the low concentration second solution, water flows into the cell and the cell expands. This is called a hypotonic solution. We see this knowledge being used in medical clinics when patients need hydration. The nurses will give the patient a saline solution which has a higher concentration than pure water. This is important because pure water is hypotonic to the cells of our bodies. If the patient were to be injected with pure water, the water would flood the cells and they would burst. When the cell is placed in the high concentration third solution, the cell shrinks in size as water flows out of the cell into the solution. This is called a hypertonic solution. This is always the case for solutions which have solutes which cannot cross the cell membrane. However, some solutes are permeable to the membrane. For example, urea and water are highly permeable, and glucose will slowly penetrate or cross the membrane. When talking about tonicity, we only look at the non-penetrating solutes, or the solutes that do not cross the cell membrane. Usually the penetrating solutes will reach equilibrium and not affect tonicity. Think about how these concepts of osmolarity and tonicity work in the body. How do the extracellular fluid, or ECF, and the intracellular fluid, or ICF, interact with each other? Pause the video to look at this chart, which gives examples of how the volume and osmolarity of our ECF can be affected in different ways. Think through each box and while using the knowledge you just gained about osmolarity, explain to yourself or to a friend why each change would take place.